Hi. Um, I am going to go over these notes with you. I know that we went over them last time, but this is really important information, and I would like you to watch this video and do it again. Okay? The last time I saw you, we talked about the structure of a virus. Okay? They are very, very simple. Remember how we talked about how um, prokaryotes, and then we have eukaryotes, and they get more complicated, like complexity, right? Eukaryotes are a lot more complex than prokaryotes. Viruses are even simpler, right? And it's also true for size. I know we talked about that last time too. So viruses are the smallest. Prokaryotes are kind of in the middle. They're still much bigger than viruses. And then eukaryotes are on the end over here. Okay? All right, here is a virus. They can look a lot of different ways. I want you to write at the top notes. Today is 2/22. Okay. Okay. So the outside part is the envelope. Okay. And it protects the outside. The protein coat is also for protection. Okay. Sorry, guys. Protein coat. Okay. So pretty simple. They have two layers of protein and it protects them. That's it. Then on the inside, we have nucleic acids. And the two nucleic acids that we've talked about this year are DNA and RNA. Okay? And then all these things, these weird things sticking out on the outside, those are all receptors. Okay? So the envelope, this might be a little different. If it's if you think it matters, but then go ahead and make some changes to yours. But I think it's everything I think it's all pretty the same. The envelope is an external protein layer. External protein layer covering the protein coat, also known as the capsid, and which might have surface markers. It helps viruses avoid the host immune system. So, right now, we are saying receptors are the same thing as surface markers. Okay, so those things are the same, receptors, surface markers. And we've already called the protein coat a couple of different things. Okay, we said the envelope is on the very outside and it covers the protein coat, which we can also call the capsid. Okay, so in biology, we like to call everything by a million names. So just know that those are all the same thing. You're going to see both of them on a test. Then we have the protein coat and capsid, right? You can see that those are the same. They have them right next to each other here. I'm going to highlight that. You guys know I love to highlight. Receptors, surface markers, protein coat, capsid. Okay. The protein coat or the capsid, it surrounds and directly protects the genetic material. DNA or RNA of a virus. Okay, so nucleic acids, This is still the same thing. We've been talking about this all year. Where can I put this? Let's put it right here. Nucleic acid equals genetic material equals RNA and DNA. Okay? Then we come down to the nucleic acid. So we have nucleic acid. It's the virus genetic material in the form of DNA or RNA, which gives directions to the host cell to create more viruses. And then these receptors, we already said that they can also be called surface markers. The receptors are spikes or protrusions or bumps, which allow a virus to attach itself to a specific cell, like a lock and a key. And that's what you guys saw in that Edpuzzle. So I'm going to say, remember Edpuzzle? If you haven't done that Edpuzzle, you can still do it. Okay, it is for a grade, so make sure you get that done. I think most of you guys did that. Okay. So we have a lot of things here that are the same thing, right? Protrusions, receptors, bumps, okay, key, lock and key, okay. Also, I know that you guys all of us lived through this pandemic, and it was caused by a virus, and it had spikes on it. It was called coronavirus because the spikes happened to look like a crown. So corona means crown. Okay. All right. This picture right here is how some viruses attach to the host cell using surface markers or spikes. Okay, so here's the host cell. It's very large compared to this virus. And then here is, remember when we talked about enzymes and we said it was like a lock into a key? It's the same thing here. Isn't biology cool how we just keep talking about the same kind of concepts? So these receptors, you can't see that, they have like a little thing like this, okay? And then right here, this virus has these little round spikes, and they literally do fit in there like a key and a lock. That's why we don't get sick from our dogs and cats viruses, because they have different viruses that fit on their cells. We have human viruses that fit on our cells. Pretty cool. Okay, let's practice these. I want you to do these. I'll read them, but I'm going to want you to do these, and then we'll check them at the end, okay? What part of the virus directly protects the nucleic acid, DNA or RNA, of a virus? Flagellum, protein coat, capsid, plasma membrane, or envelope? You can find that answer up in the top. What part of the virus has surface markers attached to it? Protein coat, capsid, plasma membrane, envelope, and nucleic acid. I know that they keep saying plasma membrane, and I hope that you remember that is the same thing as a cell membrane. Okay? That's why we don't call these things on viruses, we don't call them plasma membrane or cell membrane. We call them protein coat and capsid because they are not cells, right? They're not alive. Okay. So answer that question here. What part of the virus has the surface markers on it? Both viruses and prokaryotes contain what? Ribosomes, nucleic acids, a protein coat, a plasma membrane. See the other side if you have questions. Which of these is the best description of a virus? A non-cellular living organism, one of the smallest bacteria known, a cell at the boundary between living and non-living things, chemical complexes of RNA or DNA protected by protein. It's asking for the best description of a virus. The innermost portion of a virus's structure is made up of A, a membranous envelope, B, both DNA and RNA, C, either DNA or RNA, or D, a protein capsid. Okay, y'all, this is something I don't do very often, but we're going to draw, okay? We're going to draw a virus. Draw and label a virus. Okay, so the first thing we're going to do here, and remember guys, this is your notes. It doesn't have to be perfect. I know some of us really want it to be, but it doesn't have to be. So the first thing I'm going to do is I'm going to make a little dotted line, and that's going to be like my main shape of my virus. Okay, so can you see how I kind of made like a circle, but I did it in a dotted line? All right. Then we are going to make some mean looking eyes. Why are we making this virus look so mean? Because they make us sick. They're bad guys. Okay, that's all you got to do. Then we're going to make, bring those down a little. We're going to make it like this. Okay. So we're giving him some real mean looking eyes because viruses make us sick. They are kind of mean. They're not really mean. They're just trying to survive and pass on their genes like every other living or every other thing. They're not even living things. Okay, and then I want to make sure you guys remember that there is DNA or genetic material or RNA inside a virus. So we're going to make him have like a frowny face, but we're going to make that DNA. Here's how you do that. You're going to go like this, okay? And then you're going to take a line and just go up and down. Okay, that's how I make DNA. As long as you know what it is, it's okay. Then you're going to draw these. Remember when we studied the structure of DNA? These are the rungs of the ladder, right? Doesn't he look grumpy and angry? Okay. Then we're going to put some little spots on this guy. Some of them are filled in. Some of them are not. Or not filled in, but complete. You can always add more later. Okay. So we have what's the start of a protein coat. We have some genetic material. Okay. Now we need some receptors on the outside. Okay. So we're going to do this. Okay. Can make some tall and some not as tall. And then you're going to kind of connect them like this. Okay. Okay, and then over here, I can kind of fill that in. Okay, so here's our virus. And now we're going to label him, okay? So this is the protein coat. / capsid. Okay, and I know that sometimes it said the envelope. I wonder if the protein coat can also have spikes on it, which might have surface markers. I think that maybe the protein coat would be like inside here. Why don't we do that? Okay, and so now we're pointing to this line inside. That's the protein coat in the capsid. This on the outside then would be the envelope. Okay. These little guys are surface markers. Does it call it anything else over here? Receptors. So we can say surface markers / receptors. Okay. And then this in here, this is our DNA or RNA, which is the same thing as saying genetic material. Which is the same thing as saying nucleic acids. All right. So please notice we didn't say anything about like cytoplasm or cell membrane or anything like that because viruses are not cells. Viruses are not cells. Okay. All right. That's all. I'm going to put a key up so that you can check these. Okay. And then look on Schoology for what to do next.